So quick review from the last part of the notes. We went over what a virus is, how it's not a cell, how it's parasitic, and it contains a capsid and something inside, either RNA or DNA. They have some characteristics of living things like those we just talked about, like the DNA or the RNA, and they do evolve and replicate. But, in big red highlighter, they can only replicate using the host cell's machinery. Okay, they're non-living. They don't metabolize anything, they don't respond to stimuli, and they don't fossilize. So these things are not alive. There's some pictures, we went over some of those. They're categorized by three things: the size and shape, what they look like, what type of nucleic acid is in the inside, right? Is it RNA or DNA? And then the presence or absence of the outer envelope. So does it have on clothes or not? So if it doesn't have on clothes on the outside, it's called a naked virus. And if it does have clothes on per se, it's an enveloped virus. We talked about it being parasitic. It must infect a living cell. Therefore, it hijacks the protein synthesis machinery to replicate whatever nucleic acid it has on the inside and any other parts that it needs. Okay, sometimes they actually end up killing the cell, or the cells are damaged by a virus. Some cells are specific to organisms or a type of tissue. Those are some examples there. They usually land like a spaceship on the outside of the cell in almost this lock and key manner with these receptors. Well, some medicines we talked about mess with that manner. Look, I just realized that I use texting right here. Sorry. Should be with. And then we went through the reproduction of a virus. There are 5 steps, okay, and there are little asterisks because this is super important, okay? Super important. You have the attachment. You have the penetration. So does it engulf the virus? Does the cell engulf the virus through endocytosis? Remember that? Or does the virus inject its genome? And then we have number 3, biosynthesis. How does it synthesize the stuff? How does it mature? And then number 5, the release. Okay, does it exit through lysis or does it exit through budding? Like pinching Play-Doh off, okay? And that's where we stopped from the last notes. So we're going to finish out section 1 today. So we're going to go over first the reproduction of bacteriophages. A bacteriophage is, this is an important word. It's a big, bold word in your book. These are viruses that infect bacteria only, okay? Only for bacteria, hence the it's in the name, okay? Viruses for, I'm going to put for. So the lytic cycle, remember we're talking about reproduction here. How does these things replicate? The lytic cycle, which is also important, is 5 steps that occur immediately. The cell is lysed, and the end of the lysosome, it's lysed by the lysosome at the end. So almost the cell's like, hey guys, we can't make it anymore. And this thing, it's almost like a lysosome, is why I say that. It's actually something different that will actually tell the cell, it'll rupture the cell and tell the cell almost that, just like a lysosome, like you're done, kill yourself off so that maybe the host can survive. I guess it's almost like waving the white flag. The bacterial cell dies and hundreds of new viruses are released. So really, when I put pregnant spider up here, that's the lytic cycle, okay? The other one is the lysogenic cycle. So this one lays dormant and waits. So this is not immediate, okay? Not actively replicating. So it's almost like the virus is just waiting to happen, or waiting to for the right time anyway. So they have an extra step in these 5 reproduction steps, okay? That extra step is called integration. That's the difference. So they have 1, 2, like 2A, then 3, 4, 5. They have an extra step in there. So what is integration? Integration, I don't want it to be the same color because I don't want you to think it's another cycle. Integration is a bold term. It's viral DNA becomes incorporated into the bacterial DNA. So I think you'll see a picture of it in a minute, but it's like if this was your piece of DNA, because remember they're circular. It would be like if it went to this. That's integration, okay? That's my little example over there. That's integrating itself into its DNA. It's almost like a spy or something, like it hides itself. The lytic cycle is like coming in with like automatic, semi-automatic rifles or whatever those things are called, I don't even know, and just like knocking people dead. The lysogenic cycle is sneaky. Okay, so here is a picture from not our book, but from one of the books. So let's start with number 1 and see if we can figure out what this means. So first off, I can see that there's the lytic cycle on this side, and there's a lysogenic cycle on this side. And this little shape right here is the cell, okay, the bacteria cell. This little guy right here, that's that virus, okay? So it says number 1, the phage attaches, that means the bacteriophage, attaches to the host cell and injects the DNA. So remember, we talked about the different ways that it can go in, get its DNA into the cell. So it injects it into the DNA. It injects its DNA into the bacterial cell, okay? So it also shows you where the bacterial chromosome is. So let's move on. Okay, so go down the line here. So we have 2 different pieces of DNA. We're going to follow the lytic cycle first, okay? So at this point, we can go either way. We're going with the red. Okay, so new DNA and proteins are synthesized and assembled into virions. Okay, so it's synthesized and replicated. So all this stuff is synthesized. Remember, that was part of it, right? Using all of that, the cell's machinery to synthesize their proteins. So the next step is, how does it get out? The cell lyses, and they're all released. See the cell, it busted. And then this new virus right here can come and inject into another cell. Okay, so we're going to keep going. So it injects into another cell. There's too many colors on this thing. I can't pick, maybe red. It injects the DNA into the bacterial cell. There's 2 different DNAs here, and then we're going to go with the blue, the lysogenic cycle. So what happens here? So this is different. Do you see the difference in these 2 guys? These 2 guys are different, okay? It injected itself into the DNA. And it reproduces, the bacteria reproduces normally. So guess what? Every cell has this virus in it now. Every single one of them. So many, many, many cell divisions. And it can stay, I'll delete this, but it can stay in this lysogenic cycle as long as it needs to. It's integrating itself over and over and over. I mean, multiple cells are replicating in that virus. The virus DNA is being passed on for a long time until one day one of them is going to go this way. Okay? Going to go the dark side. That's what happens. So lysogenic cells carry copy of the genome in the future induced into lytic cycle by environmental factors. So I just said that. These lysogenic cells up here carry the genome and will go into the lytic cycle at some point and will be induced into that, probably by some sort of environmental factor. For example, this is a good one, fun fact. Bacteria that cause strep throat. So bacteria causes strep throat, okay? Is carrying, it's in 3B, it's carrying that it's integrated with the bad DNA, like we talked about earlier. This is strep throat. If it has the green in it, then it becomes scarlet fever. If this happens. Is that not crazy? So if you have scarlet fever, or if you've ever had it, or if you know somebody that's had it, that means that not only is their body infected with bacteria that causes it, that bacteria is also sick. So it's like the bacteria is sick with the virus, and the bacteria infects your body or that person's body. So they're almost the same thing. I just thought that was cool. So the reproduction of animal viruses. So we're only talking about animals now. So they introduce genetic material differently. Some envelope viruses, they attach in the fusion of the spiked envelope. So some of them are going to be enveloped. Remember, that means they have clothes on. Many naked and other envelope viruses use endocytosis, enters uncoated, and then one of the cycles happen. It says some viruses enter directly into the lytic cycle, causing rapid and severe destruction of host cells. So think about it. If a virus enters into the lytic cycle and it causes rapid and severe destruction, what do you think that's going to happen in your body? Your body's like, oh my God, get rid of this stuff, and it's going to make you throw up and do other bad things we're not going to talk about. I mean, not bad things. It's not bad, but gross stuff. So here's a one that's talked about. HIV enters the lysogenic cycle and is inactive for many years before it turns into AIDS. So HIV is AIDS. But HIV it's when it's in the lysogenic cycle. AIDS is when it turns into the lytic cycle. Okay, so I hope that makes some sense for you. There's tons of medicines that try to keep that virus on this side. I'm going to embarrass myself, but I think it's Magic Johnson maybe that has HIV. I'm not 100% sure on that, but I don't keep up with stuff like that. But if it is him, then a lot of doctors have helped him stay over here, because when it goes to the lytic cycle, it's bad news. Destroys everything. You don't live very long when you get to that side. So how do these, the reproduction of animal viruses, how do these release? So some release by budding. It's almost like this is, budding is like picking Play-Doh. Like, you know how you pinch off Play-Doh? That's what budding is like. Picks up its envelope from the plasma membrane. It just takes part of the plasma membrane of that cell and uses it as its own. But naked viruses release, so the ones that don't have anything outside, released by lysis of the host cell. So that was like that bursted cell, okay? And the guts are coming out. Okay, so that's all about, we talked about bacteriophages, the ones that are specific to bacterial cells. We talked about some animal viruses and how they work. Now we're going to talk about retroviruses. Retroviruses. So what is a retrovirus? It is, this is the definition, animal viruses with an RNA genome. We've only talked about DNA thus far. That have to be converted to DNA first by the host cell by an enzyme called reverse transcriptase. That's a big one. Know what this is. And I always know if you're paying attention to me when I talk on these things, or if you've even watched them, by your tests. So, you know, when I say, hey, this is going to be on the test, and then you miss it, then you're probably not paying much attention to me. So usually when we put in or we injected the DNA from above, it was already in DNA form. So we didn't have to do anything. Because remember, DNA is read by RNA. Remember that? mRNA. And then it's made into, it's read for proteins. Remember, this is a central dogma. But what happens if I come in at this point? I have to be reverted back so that I can start over. Okay? I have to go from RNA to DNA. That's what retroviruses do. That's what these guys do right here. Who does it? Reverse transcriptase. Because remember, DNA to RNA is called transcription. So, of course, whoever named this named it almost reverse transcription. Except it has ASE on the end, so that means it's an enzyme. So there's a little thing called cDNA in the middle, but you don't have to know that. That helps this process out. So let's look at this picture. All right, so step one, we have attachment. Remember, we're talking about how these things work. Step two, penetration. This one was engulfed. Then this guy's, because this is RNA, this guy's step three is called reverse transcription. So you see this little piece of RNA right here. It's turned into this piece of DNA. Then the DNA comes into the nucleus and integrates into the cell's DNA. Then it's read into mRNA. I'm sorry, I skipped circle on that one. We integrated it. I said that though. And then we make some read it, or mRNA reads it. Look at this word right here. What is this word? What chapter is that from? The ribosomes are reading this piece of mRNA, which has the bad DNA in it, and then this is biosynthesis happening. And then these little guys are going to shoot out and make more. So AIDS is an actual retrovirus because remember, it's an animal virus that actually has RNA. So you learned something. If you learned anything at all, you learned that AIDS is a retro. My pen's going dead on me. Come on, pen. Oh, well. I don't think it'll let me do it with my finger. It won't. Oh, well. Well, anyways, you learned today that AIDS has RNA and it's a retrovirus. So some drugs will delay some of these things. So the drug called AZT is a reverse transcriptase inhibitor. So this guy right here I actually covered it up. That little blue dot right there that says reverse transcriptase. The medicine AZT inhibits this so that this process cannot happen, or it slows it down anyway for people that have HIV or AIDS. Acyclovir inhibits the replication of HIV viral DNA. So if you get cold sores and things like that too, people will prescribe you that. Somebody that's absent and is watching this is going to be like, why did she write age-R on this sheet? I think we're almost done. We have 3 more. All right, so there's some emerging viruses out there. This used to not be a problem. Now it is. AIDS, the AIDS outbreak, West Nile virus, HPS, SARS, the bird flu. We could go on and on, right? Could add COVID to this. This is why it's important to get the flu shot every year, because these viruses are evolving every year because, like I said, their job is to survive. They want to survive too. And HIV is also rapidly emerging. That's why we can't find a cure for it. It's almost like they emerge before we can determine how to stop them. So what are viroids? 2 more slides. There are over 1,000 different viruses that cause disease in plants. So on your notes, you need to put viroids or viruses for plants if you want to. There are 12 of those that aren't actually viruses, but they're called viroids. And what an actual viroid is it's for plants, but it's like a naked strand of RNA. There's nothing else there. Last slide is prions. These are proteins that normally exist in an animal, but have different conformations or structures. If they change shape, so if like a protein is supposed to be shaped like, I'm just going to make it up, like a circle, you're good to go. But if that protein breaks down over time and it's almost like losing your mind, that protein changes its shape to like a triangle, then it's really, really bad. Like we talk about conformational change in shape, because shapes are so important in organisms because of all those lock and key manners. So when you change something's shape, it can be detrimental. It can cause fatal brain disease, like this was mad cow disease, neurodegenerative diseases that destroy nerve tissue and brains. These are untreatable. There's nothing you can do about it at this point. So one of the last chapters we talked about, or I talked about rather, when we talked about Miss Brown just getting too old to teach. When Miss Brown's hitting 85, like I hope somebody comes in and walks her out. I hope somebody's walked me out at that point too, because we don't want to turn into a prion. We don't want to do something that's untreatable or fatal. Same thing goes for here. So that's why your bodies and organisms have mechanisms to, kill things in your body or degrade something and almost, put it into retirement, because when the older you get, it's like, older people driving. Sometimes it's just time to take away the keys.